conversations about the state of ocean acidification science in the Southeast region. These webinars are intended to create a dialogue among scientists to identify what is known, what isn't, and what research in other regions of the United States can be applied to better understand ocean acidification and its impacts in this region. It's hoped that with this information, webinar participants will be able to collaborate to better understand and adapt to ocean acidification moving forward. I'm Paula Keener with NOAA's Office of Ocean Exploration and Research and a member of the SOCAN Steering Committee, and I'll be moderating today's session. Jennifer Bennett-Mintz, Education and Outreach Coordinator for NOAA's Ocean Acidification Program, is also on the line with me today and will be helping to facilitate today's session. During the presentation, all audience members will be in listen-only mode. You're welcome to type questions related to technical issues or questions for the presenter into the questions box at the bottom of the GoToMeeting panel on the right-hand corner of your screen. Jennifer and I will be moderating incoming questions, and we will respond to them or pose them to our speaker. We are also recording this session, and a video recording and a PDF of the presentation will be available on the SOCAN website. We are very excited to welcome our speaker, Dr. Kim Yates. Kim is a senior research scientist at the U.S. Geological Survey Coastal and Marine Science Center in St. Petersburg, Florida. Her research currently focuses on the effect of ocean acidification on coral reef health and growth, estuarine ecosystems, and natural ocean acidification refuges for marine organisms. So Kim, I'm going to hand the presentation over to you.
Okay, I think I have it. Can you hear me, Paula?
Roger that.
Okay. Thanks everyone for joining today, and thank you to SOCAN for giving me the opportunity to talk to you about coral reefs in our region. And as soon as I can get this to activate, we will move forward. Okay, there we go. I have a lot of material to cover for you guys today, and so I'm going to start out by giving you a brief overview of coral reef basics that are relevant for our discussion of ocean acidification today. And then I want to spend a little bit of time setting the stage and talking about the current threats to coral reefs and the existing baseline of reef health that ocean acidification is impacting in our region. Then I'll talk a little bit about the effects of ocean acidification on coral reef community metabolism and some of the long-term implications for coral reefs and specifically reef structure. And then I'll go through some results from some recent studies on ecosystem level responses of coral reefs to ocean acidification in our region. Coral reefs provide a number of ecosystem services. Many of these, most of you are probably familiar with. Some of them are not so obvious. First and foremost, coral reefs provide fish habitat for one of the most biologically diverse ecosystems in the ocean. They also provide a major source of food, especially for some of our developing nations in the Caribbean. Reefs serve as a natural barrier along the coastline and actually protect coastlines from storm waves and tsunamis. As reefs grow,
the broadcast is now starting. All attendees are in listen-only mode.
Paula, I have a question for you. I just said that the broadcast is now starting, so have I started this too soon? Do I need to back up?
Kimberly, Jen, we're getting no audio.
Okay,
yeah, so Kim, go ahead and back up if you wouldn't mind. Yep.
Okay, so I'm assuming everyone missed the introductory remarks. Is that correct?
It is.
Okay, so let's start over. Sorry about that, gang. All right, so this is the outline for our talk today. I have a lot of material to cover, and I apologize if some of you have already heard this on audio. I'm going to begin by giving you an overview of some of the coral reef basics, Coral Reefs 101, that are relevant to our discussion of ocean acidification today. And then I want to spend a little bit of time sort of setting the stage and talking about current threats to coral reefs and the existing baseline of reef health that ocean acidification is affecting in our region. Then I'll talk about the effects of ocean acidification on coral reef community metabolism and what that means with respect to long-term implications for both coral reefs and coral reef structure. And then I'll go through some results from some recent studies on ecosystem level responses of coral reefs to ocean acidification. So coral reefs provide a variety of ecosystem services. Some of these you are familiar with, and some of these may not be so obvious. First and foremost, coral reefs provide fish habitat for one of the most biologically diverse ecosystems in the ocean. They're also a major source of food, especially for some of our developing nations in the Caribbean. Coral reefs serve as natural barriers along the coastline and actually protect coastlines from storm waves and tsunamis. And as coral reefs grow and carbonate structures degrade, they actually produce sand for many of our beaches. Coral reefs provide a number of pharmaceuticals, such as anti-inflammatories, some pain medications, and anti-carcinogens. And then we all know that they're a source of many recreational activities, such as fishing and diving. And corals also provide a mechanism for long-term storage of carbon through production of calcium carbonate. These ecosystem services support a number of different jobs and businesses that are at risk in our region from ocean acidification. This information actually comes from a pamphlet that was put together by the Ocean Conservancy for a recent Florida Ocean Acidification Roundtable meeting that was held at Mote Marine in Sarasota in September of this year. So recreational coral reef diving, fishing, and viewing of coral reefs generates about $60 billion in local sales in our state, and supports about $2.8 billion in local income. That supports about 70,000 jobs in Florida, and this also supports a long line of what we call downstream industries. So for example, marinas, hotels, restaurants, gift shops, and other businesses that are along coral reef coastlines. All of these ecosystem services depend on the ability of coral reefs to produce these three-dimensional structures. This is a very generalized diagram of coral calcification. This is a coral polyp, and at the base of these polyps there are very specialized cells called calicoblastic cells. Between these cells, or the outer layer of this tissue, there's a small fluid between the cell, the animal, and the carbonate basal plate that it sits on. And this is where the calcium carbonate is actually produced. These cells actually pump the chemical constituents into this space that are required for development of the carbonate mineral structure. And you can see that carbonate combines with calcium to form calcium carbonate, which forms that mineral skeleton. And this process very much depends on the production and pumping of both hydrogen ions and carbonate ions that are both affected by ocean acidification. So it's this coral calcification process that creates this three-dimensional structure of coral reefs. For some reason my buttons are not responding to changes. There we go. All right, this calcification process is a reversible process. So this is a balanced equation of the reaction for calcification. There's our carbonate, there's our calcium, and we create calcium carbonate. But this reaction can be reversed so that you actually get carbonate dissolution. So if you add CO2 to the reaction and water, it can cause calcium carbonate to dissolve back into its constituents. And you can also get dissolution of carbonates by decreasing carbonate ion concentration or mineral saturation state, which is based on carbonate ion concentration. Coral reefs form very slowly over thousands of years through this process of biogenic calcifications. As marine organisms calcify, they create these massive structures of carbonate skeletons. Over time, as the organisms grow and die, these skeletons break down and produce carbonate sediment. This carbonate sediment fills in the reef framework and eventually becomes cemented, and that allows the upward vertical accretion of coral reefs. So that's how they grow. In 1988, Budemeyer and Smith summarized reef accretion rates from a variety of different papers and methods and compared those to sea level rise. So for example, they looked at linear growth rates of individual coral species. They looked at accumulation rates of reefs from the post-glacial Holocene in the geologic record. And then they also looked at calcification rates using a chemical proxy that we call the alkalinity anomaly technique, or alkalinity depression. And you can see that during this time, reefs were generally keeping up with rising sea level. This is very important. Shallow water reefs have to keep up with rising sea level because they contain photosynthetic organisms in their tissues and so need to stay where they can reach enough light to help them grow. But keeping up with rising sea level depends very much on this balance between calcification and chemical dissolution of carbonates. We also call this chemical erosion. It also depends on other types of erosion in addition to chemical erosion, like physical erosion and biological erosion, and how much sediment is exported from the system. Depends on coral reef accumulation rates. They have to be accumulating fast enough to keep up with the rise of sea level. And so the bottom line, the end game, is that calcification and accumulation has to exceed erosion and transport in order for these reefs to keep up. And this very much depends on healthy corals and other calcifying organisms on the reef. So we all know that coral reefs around the world are threatened. This is a picture of a coral head from Grecian rocks in the Florida Keys in 1961. This is the same coral head in 1988 and again in 2001. So you can see that over time, over the last few decades, we've had an enormous amount of loss of reef structure, and we see this transition to these algal-dominated communities. And in fact, the abundance of reef-building corals has decreased over 80% since the 1970s on Florida and Caribbean reefs. And so we're seeing this transition from these ecosystems that were dominated by calcification and are now becoming dominated by production of organic carbon. Reef degradation over the past couple decades has been attributed primarily to the local stressors, and I like to call these guys the local villains. You're probably familiar with most of these. Pollution is obvious. Overfishing causes problems, particularly overfishing of herbivorous fish that keep the algae mode back on these reefs. Terrestrial runoff of sediments is important. Not only can it bury and suffocate corals, but also it carries contaminants along with it very often. This might be overkill on this particular slide, but physical damage is important. And yes, there are some places in the world where people still use dynamite to fish on reefs, but in our area we're talking mostly about anchor damage and physical breakage due to recreational and fishing activities. All of these stressors can lead to coral disease. And then we have the global stressors, or what I like to call the global villains. And these have really been considered emerging threats. And what we're talking about is those threats caused by elevated atmospheric CO2. And for the chemists in the crowd, you might recognize these little guys as CO2 molecules. So these are our global villains for the story today. So we're talking about global warming and ocean acidification. The IPCC AR5 report projects that 33 to 66% of the world's coral reefs are subject to long-term degradation in the next few decades due only to climate-driven impacts, solar or thermal stress, meaning elevated temperature, light, and UV radiation, and ocean acidification. So a little bit on thermal stress or global warming basics. If water gets about 1 or 2 degrees Celsius higher than summertime averages, then corals can get stressed and bleach. This is a picture down here showing a healthy coral that has its algal symbionts with it, and when they bleach, they expel their symbionts, and you get this translucent tissue color that looks white because you can see the underlying carbonate skeleton. Elevated light and UV can actually increase thermal stress and bleaching, and this is a graph from some work by Derek Manzello published in 2015. This shows mean seawater temperature from 1987 through 2014. This red line is a thermal stress threshold that was developed at Molasses Reef from some temperature records there. And you can see that since 1987, we've had 6 major Keys-wide bleaching episodes. And between 2006 and 2013, we've actually had moderate bleaching every single year except for 1. And so annual bleaching is expected to occur as early as 2020 in the Florida Keys. High temperatures and bleaching also can leave corals more vulnerable to disease. This is a picture from Marilyn Brandt at the, well, she's at the University of Virgin Islands now. She was in Miami. This shows the stages of both bleaching and then coral disease, the progression over about 1 month in 2005. So you can see that the coral starts bleaching and disease sets in because it has become much more vulnerable to disease. So this can very quickly kill part or all of the coral colony. By 1998, 82% of the coral monitoring stations, there were about 160 in the Florida Keys, were affected by coral disease. Bioerosion also causes loss of reef structure. Once these corals become susceptible to disease and bleaching and begin to die, these areas of dead coral are much more vulnerable to bioerosion. And that's when various animals and microorganisms can actually go in and eat away or wear away at the coral's limestone structure and their tissues. This results both in loss of reef structure and function. So here's another picture from 1959 of a coral head in the Coral Keys and the same coral head in 1998. And this is very much what the current baseline of reef health looks like in most areas in the Atlantic and Caribbean. So this is what we're starting with. We see these transitions from these coral-dominated communities to communities dominated very much by production of organic carbon and much less calcification. Because of this, one of the ways that we can measure reef health is in terms of what we call coral reef community metabolism. And so this is that balance that we're talking about between net ecosystem production or organic carbon gains and net ecosystem calcification or inorganic carbon gains. And so what we're talking about is the balance between what we call primary reef functions. So photosynthesis, respiration, calcification, and dissolution. So the question that we want to come back to then is, how is ocean acidification anticipated to impact coral reef community metabolism? Many of you who have been joining our webinars have probably seen a slide like this showing ocean acidification basics. I'm going to walk through this again and specifically point out what's relevant for our discussion with respect to coral specifically. So ocean acidification occurs as elevated atmospheric CO2 equilibrates with the surface water of the ocean. CO2 then combines with water and produces carbonic acid. That causes a decrease in ocean pH. Carbonic acid can also further dissociate into bicarbonate ions and carbonate ions, and these are what we call the primary dissolved inorganic carbon species in the ocean. This dissociation reaction is very much pH dependent, so at higher pH you have carbonate ions, and remember that's one of the things that's really important for coral calcification. But as pH decreases, you get a decrease in carbonate ion concentration and you get more bicarbonate and carbonic acid. This system is relatively easy to characterize. We can measure two of what we call 4 carbonate system parameters. So we can measure one of these or 2 of these 4 parameters and calculate this entire speciation and much of the chemistry associated with ocean acidification. And in fact, there's a growing body of literature that shows that coral reef community calcification rates are directly dependent on carbonate ion concentration. This is one of the classic graphs from some early work by Chris Langdon that was done in the Biosphere 2. You can see community calcification rates on this axis, and you can see decreasing carbonate ion concentrations here, and you get a linear decrease in calcification rate with carbonate ion concentration. The 0 line is here, if you can see my pointer moving back and forth. So numbers less than 0 indicate dissolution. Numbers greater than 0 indicate growth. So you can see at some point we actually begin to get dissolution as carbonate ion concentrations decrease. And so that means that we actually have a lot of what we call OA losers on the reef. So not just corals, but other things like calcareous algae, forams, various echinoderms, and shellfish are susceptible to this as well. So calcium carbonate production rates are actually estimated to decrease by as much as 60% by mid-century. Ocean acidification also increases carbonate dissolution, and there's actually kind of a double whammy effect going on here. So calcification itself, this is the reaction that I showed you earlier, actually produces CO2, decreases carbonate ion concentration, and decreases pH. So coral calcification alone can actually exacerbate ocean acidification conditions. And it also causes carbonate dissolution. Again, this is that reverse reaction. So this can be bad for corals if they're subject to dissolution. However, if you get dissolution of carbonate sediments, you can actually consume the CO2 in the reaction, that's there, increase carbonate ion concentration, and you can actually buffer pH. So again, we have some losers in this situation, but carbonate dissolution may also help buffer pH in some instances. OA can also increase photosynthesis. This is the photosynthesis reaction. You have CO2 and water, and you get a little bit of sugar and oxygen. And Palacios and Zimmerman in 2007 showed that seagrasses actually increase photosynthesis and reduce light requirements with increased PCO2 in the water. And for macroalgae, they actually show a 52% increase in growth at 2 times atmospheric CO2. And so we actually have some winners with respect to ocean acidification. So things like various types of seagrasses and algae will actually benefit from ocean acidification. Ocean acidification also increases susceptibility to coral bleaching. This is a slide that is courtesy of Aaron Mueller at Mote Marine, some work done by Ken Anthony et al. In 2008, showing that bleaching actually increases with ocean acidification, both for coral species and coral and algae. However, some work by Aaron Mueller shows that low pH can actually reduce the progression of black band disease. So you get fewer lesion or slower lesion progress at lower pH in certain coral species. And it also reduces the virulence of coral and algae disease as well. And this is some more work by Aaron Mueller. So with ocean acidification, we may actually see an increase in coral bleaching, but a decrease in infectious disease in both coral and algae and corals. And so we're not really sure at this point whether or not there will be an increase in infectious disease as coral bleaching increases in the future with respect to ocean acidification. Ocean acidification also increases rates of bioerosion. This is some work by Anne Triboulet in 2009, and at 2 times pre-industrial atmospheric CO2, eudendolithic algae actually dissolve about 50% more carbonate. So the long-term implications for the future of both reef function and structure are probably beginning to become apparent. We're going to see a lot less reefs that looks like this and a lot more reefs that look like this. We're getting this transition from dominance of calcification to production of organic carbon as the dominant process on these reefs. So one of the ways that we try to quantify these effects is we use what we call an integrated seascape approach or an integrated reefscape approach in this particular circumstance to look at the impacts of climate change on ocean acidification and coral reefs. So what we do is we integrate and synchronize research on the chemistry, the physical environment and processes, and the biology and biogeochemical cycling on reefs to try to get an ecosystem level picture of how ocean acidification is affecting these systems. This is one example of some of the work that we've been doing over the past several years. We've been using this approach to measure community level calcification and dissolution rates on coral reefs. We combine things like mesocosm studies using these chambers that sit on the seafloor. These are actually about the size of a large full-size pickup truck. We use these kinds of chambers to trap water over the seafloor, over various kinds of coral communities and associated communities, and then measure changes in the water chemistry over time. And then we can use a lot of those chemical parameters as proxies to calculate things like community calcification and dissolution and photosynthesis and respiration. And then we combine that with long-term monitoring of the chemical environment so that we can look at how these biological processes change in response to the chemical environment. We've done this type of work at a variety of different study sites in the Atlantic, Pacific, and Caribbean, including in the Florida Keys, U.S. Virgin Islands, Puerto Rico, Florida Bay, and also in Hawaii. And this is an example of what some of that kind of data look like. So if you look at this bottom graph, you can see PCO2 as the blue line and total alkalinity as the red line. And that's one of the chemical proxies that we use for calcification. So you can see that as PCO2 increases, usually during the night, you also see an increase in total alkalinity. And increasing total alkalinity is an indication of dissolution of carbonates. And as PCO2 decreases during the day, you also see a decrease in total alkalinity as you're getting calcification. This red line here indicates a little over 500 microatmospheres of PCO2 in the water, and so you can see that during the night, the water column actually gets elevated well above 500 microatmospheres of PCO2. So if you look at these numbers on the data points on the total alkalinity line in red, those correspond to the calcification rates calculated from the alkalinity values on the top graph on the left in the red line here. And again, you can see that during the day, we get net production of carbonate above the zero line as positive numbers, and during the night, we get dissolution of calcium carbonate indicated by the negative numbers below the line. So we can use this type of data to calculate what we call dissolution thresholds. So if we plot rates of calcification versus PCO2 and carbonate ion concentration, we can begin to tease out what we call PCO2 and carbonate ion thresholds for dissolution. So again, here's the zero line, and positive numbers indicate calcification, negative numbers indicate dissolution. And you can see dissolution begins happening, in this case, at over 500 microatmospheres of PCO2 and at about 181 micromoles of carbonate ion concentration. So dissolution occurs when the seawater PCO2 is greater than the PCO2 threshold, and the carbonate ion concentration is less than the carbonate ion concentration threshold. And if we look at the dissolution thresholds in the areas of our study, here's PCO2, here's carbonate ion concentration, you can see that they're pretty variable. This is the average of all of the study sites that we've been working in, and this variation is due to things like community composition, seasonal variations in calcification rates, different sediment compositions with respect to the carbonate minerals especially, biological control on calcification, and also water mass residence time. So in this particular instance, though, we're showing that on average, the PCO2 threshold for these study sites is about 584 microatmospheres and carbonate ion concentration of 208 micromoles, and that corresponds to a saturation state of about 3.1. So even though these are variable, these dissolution thresholds are variable, if you begin to plot these kinds of thresholds from a variety of places around the world, you can see that there is some merging evidence for dissolution thresholds. This graph shows aragonite saturation state in this red line, predicted out to 2100. That's some data from Joni Klapis. And if you plot our average threshold, which is this black dot right here, and also this green dot, which represents the threshold calculated from a modeling study of about 9,000 different reef locations around the world. Also some work from the Shiraho Reef and also work from the Biosphere 2. You can see these beginning to converge in a fairly narrow band. And that threshold range is about 3.0 to 3.2 on the saturation state, and about 520 to 600 parts per million of CO2. And so we think that this might be what we call the calcification dissolution tipping point. And this is expected to occur around the year 2050 to 2060. We can also take this kind of data and calculate reef accumulation rates and look at those relative to sea level rise, much like Budemeyer and Smith did back in 1988. So if we take our data and look at dissolution and calcification rates, you can see that in some of these places, up to 80% of the carbonate that's produced during the day is actually dissolved during the night. So there's already a very much sediment dissolution already occurring. And then if you look at those accumulation rates relative to sea level rise, which is this lower graph, these red locations are the study sites that I've been showing you. You can see that many reefs are not keeping up with sea level rise, and none of our Caribbean sites are keeping up with sea level rise. And remember that this does not take into consideration the fact that ocean acidification increases dissolution and decreases calcification. This is what's happening right now. This is some work that Chris Langdon is doing in cooperation with NOAA. And these slides are courtesy of Chris. The NOAA ocean acidification test bed that's located at Chica Rocks in the Florida Keys has been measuring water chemistry, including PCO2, since about December of 2011. So this graph shows PCO2 in the water from 2011 to 2015. And you can see that during the winter months and the fall months, CO2 actually far exceeds 500 parts per million. And if you look at calcification and dissolution at that particular site on Chica Rocks in the Florida Keys, you can see that this particular reef is already dissolving during the winter. Remember, here's your zero line. Positive numbers indicate calcification, negative numbers indicate dissolution. And so you're seeing several months where you're actually getting dissolution of that reef. And in fact, dissolution is occurring throughout the Florida Keys. This graph shows rates of calcification for reefs at these 7 locations. This is the Florida Keys, at these 7 locations throughout the Florida Keys. They correspond to these numbers here. And you can see that reefs are dissolving during the winter in all but 2 of the reefs that are located in the lower Florida Keys. Additionally, one of the confounding factors that we don't know much about at this point is that Florida is a carbonate platform that supports these reefs and the coastlines along them. So here's the Florida carbonate platform. This actually formed millions of years ago when sea level was much higher and the Florida Peninsula was covered by a shallow sea. Calcifying organisms created thick deposits of carbonate material, and that accreted into this carbonate platform. After sea level fell and the carbonate platform was exposed, karst topography, what we call karst topography, developed. In much of this Florida peninsula, the carbonate platform is covered by this karst topography, which basically are solution holes in the carbonate platform. And that creates what we call sort of a Swiss cheese subterranean geology. That karst topography is what forms most of Florida's sinkholes and aquifers and develops what we call leaky coastal margins. So karst topography and sinkholes form through this process that looks a lot like ocean acidification. So carbon dioxide combines with water in the atmosphere from rain. It falls on the carbonate that's exposed. It dissolves the carbonate to the point that it collapses and creates these sinkholes. So what we don't know is as atmospheric CO2 increases, whether or not this process will be exacerbated and actually create more sinkholes and actually increase the conduits for groundwater. This karst topography is what houses our Florida aquifer system and also houses our springs, many of which pop up off of the coastline and bring fresh water and any nutrients or contaminants that it might contain out to the reefs. So one of the things we don't know is how coastal acidification will affect these carbonate coastlines and groundwater flow, particularly to aquifers in our Florida reefs. So key points so far. Reefs were not predicted to start dissolving until about 2050 to 2060. The finding that reefs in the Florida and the Caribbean are already starting to dissolve, even if only for part of the year, is very surprising and alarming. The effects of coastal ocean acidification on carbonate platforms are unknown, but are of a particular concern because of our leaky coastal margins. And the primary point is that the threat of ocean acidification in Florida and the Caribbean is not an emerging threat. It's already here. It's not a future threat. So there's a lot of doom and gloom there. So the question is there any hope? Will reefs keep up with rising sea level, or are they going to begin to erode away and become meccas of organic carbon production? There is some hope, in fact. In fact, seagrass turns out to be one of our superheroes. Seagrass photosynthesis can actually buffer ocean acidification. Here's an equation I showed you earlier, and photosynthesis actually consumes CO2, produces oxygen, and increases seawater pH. So for organisms to benefit from, even if only for part of the year, is very surprising and alarming. The effects of coastal ocean acidification on carbonate platforms are unknown, but are of a particular concern because of our leaky coastal margins. And the primary point is that the threat of ocean acidification in Florida and the Caribbean is not an emerging threat. It's already here. It's not a future threat. So there's a lot of doom and gloom there. So the question is there any hope? Will reefs keep up with rising sea level, or are they going to begin to erode away and become meccas of organic carbon production? There is some hope, in fact. In fact, seagrass turns out to be one of our superheroes. Seagrass photosynthesis can actually buffer ocean acidification. Here's an equation I showed you earlier, and photosynthesis actually consumes CO2, produces oxygen, and increases seawater pH. So for organisms to benefit from this, it depends on proximity and water flow over these seagrass beds and also water mass residence time. So for example, if you have a seagrass bed and the water is flowing over it, it has to flow slowly enough for there to be enough time for photosynthesis to affect the overlying water chemistry and change the pH. And then any organisms that will benefit from that have to be downstream and actually feel that water mass coming over them, so downstream of the predominant current. So these guys who are sitting upstream of a seagrass bed may get the short end of the chute. They're not so happy, but these guys sitting downstream of the seagrass bed are happy as clams and actually prefer their CO2 and beer. So there's some recent studies that indicate, in fact, that some reefs may actually function as ocean acidification refuges. And in fact, right here in the Florida Keys, Derek Manzello's group showed in 2012 that the middle Florida Keys actually looks like it's serving as an ocean acidification refuge because there are a number of patch reefs that are surrounded by rings of seagrass that are photosynthesizing and locally increasing the pH and saturation state. There are also some places in the world where it looks like some reefs are acting as solar stress refuges. And for example, in Palau, there are reefs that are growing shaded in very turbid water. And this is protecting the corals from UV and irradiance stress and allowing them to grow at higher temperatures and actually producing what we think are more resistant species to thermal stress. This is some work that I've recently been doing in Hurricane Hole in St. John in the U.S. Virgin Islands. And we found actually that there are 33 species of reef-building corals living underneath and attached to mangrove prop roots in bays in Hurricane Hole. And we actually think this is serving as a natural alternative or non-reef shallow water refuge that they're protecting these corals from both solar stress and ocean acidification. These corals in the lower picture are actually growing only a few tens of meters away from these corals in the mangroves, and they are very much struggling to survive, while the corals in the mangroves are absolutely thriving. This does not happen in any mangrove habitat. Most mangrove habitats throughout the world are not suitable for coral refuges such as this. The characteristics of St. John, the physical, chemical, and biological characteristics, there's a special combination that makes this happen here. These islands have very small watersheds, the bays have very small watersheds, and there's no permanent source of fresh water inflow here. And so the water is very clean and clear here, and the biological and chemical heterogeneity is just right for producing these conditions. These graphs show dissolved oxygen on the top and pH on a nearby reef to this ecosystem. These are the gray bars, and all of the red, blue, and green bars are in the mangrove coral habitat. And you can see that dissolved oxygen is much higher in these habitats than on the reef, and pH is also much higher in these habitats than on the nearby reef. These graphs show PCO2 and pH, again on the reef in gray, and in the mangrove habitat in the other colored bars, and you can see that PCO2 is actually much lower, and actually lower than the PCO2 threshold, average PCO2 threshold in the mangrove habitats, and mineral saturation state is actually much higher than on the reef and higher than the saturation state threshold. This top graph, it's a little bit messy, but if you squint, you can see that the black, blue, and red bars on top show irradiance, or PAR, photosynthetically active radiation outside of the mangroves, but nearby. And these green bars down below show light, or PAR, underneath the mangrove canopy. And it turns out that the mangrove is actually reducing PAR by more than 70% and shading these corals. The lower graph shows temperature in the mangroves in the red lines and on the reef outside, or in the black lines, and you can see that the temperature is actually higher in these mangrove coral habitats than on the reef. However, these corals are bleaching much less, and so because they're getting shading from the mangrove canopy, which is reducing UV and light stress, they're able to live and grow at higher temperatures and not bleach than the corals on the reef. So this is actually a very unique situation. In fact, this is the first and only mangrove coral refuge that has been identified and characterized in the world. We think that there are other places in the world where this might be happening, mostly from anecdotal evidence by way of tourist photos on Google. In fact, we see evidence for it beginning to happen in Raja Ampat in Indonesia, in Bocas del Toro in Panama, and in Pelican Cay in Belize. And I recently got a report of some observations from some of my colleagues at FWRI who have seen corals starting to grow in the mangroves in Biscayne National Park in South Florida. One of the most intriguing aspects of this is that we have not been able to find any evidence anywhere in the geologic record for coexistence of live corals and mangroves. And so we think that this actually might be what we call a new or novel ecosystem transition due to unprecedented rates of recent climate change. So Mother Nature is finding ways to help itself in times of crisis. So then to briefly summarize, we find that coral reefs grow very, very slowly and are degrading rapidly in the Florida Keys and the Caribbean. And this is a particular concern because what has taken thousands and thousands of years to grow is degrading very quickly on the scale of decades. Over the past several decades, there have been a number of local threats that have already set what we call an unhealthy baseline that ocean acidification is also now affecting. So we're not starting in a good place in the Caribbean and Florida with respect to future conditions for ocean acidification. The end game is that whether it's a local stressor or a global stressor, including ocean acidification, all of these stresses cause loss of reef function and loss of reef structure. And reefs in the Keys and the Caribbean are already starting to dissolve, so the threat is already here. There are some bright spots. Again, Mother Nature is finding some ways to help itself through these natural shallow water refuges, whether they're on coral reefs or whether they're alternative or non-reef refuges. And protection of these natural refuges is one direct local action that we can take to help manage these global climate change impacts, and especially as sea level rises. So I want to leave you on a positive note with the end or possibly a new beginning as these ecosystems begin to evolve and transition over time, and I'll open the floor to questions.
Thank you so much, Kim. That was a fantastic presentation. We do have some questions, and we will start with one from Dwight Gledhill. And Dwight asked, how much of the decline in the net accretion in the Florida Keys over the industrialized period can be attributed to the loss of live coral cover versus ocean acidification?
That's a really good question. We don't know the answer specifically to that question yet, but we are on the verge of getting that. In fact, some of the data that Chris Langdon is putting together, he's actually working right now on converting that into a change in accumulation rates. And so he's, through that work, going to be able to tease out how many millimeters per year, if you will, of reef seafloor elevation that we've lost due specifically to chemical dissolution. And then we at the USGS right now are working on looking at bulk changes in reef geomorphology that we can combine with that kind of data to begin to sort of tease those things out and begin attributing those changes either to chemical dissolution or other forms of erosion or coral loss. So we're not quite there, but we're close, Dwight.
Great, thanks, Kim. And Libby Jewett wants to know, and I guess this is in addition probably to the mangroves that you just mentioned where there is coral reef growth, are there any areas of refuge that we should protect?
Certainly these types of areas, you know, this is a brand new type of ecosystem that we found, and in that 33 species of corals that are living there, there are at least 4 threatened species. This is actually within the national park, Virgin Islands National Park, and so it's already getting some protection. Whether or not we need to take additional measures in some of these places, you know, remains to be seen because they're already doing quite well. The Florida Keys certainly has these special protected areas. Not sure, maybe we need to take a look at moving some of those as we identify areas that are serving specifically as ocean acidification refuges. You know, those might be additional areas that we wanted to add some additional protection to. I hope that answers what she was trying to get at.
Okay, great, thank you. Let's see, and then Dwight also wants to know if the net accretion of the reef can be bolstered against OE through the managed increase of live coral cover.
That's an excellent question, actually. We all know that there are a variety of coral nurseries out there, and Ryan Moyer is actually doing some work in the northern Florida Keys where it looks like Acropora cervicornis is actually coming back on its own. And so, you know, certainly those are localized areas where things are doing better, and there are other areas, for example, Buck Island in St. Croix has had some success with recovery of coral. And so the bigger question is, there's no question that these areas should be protected. The bigger question is whether or not that's going to be enough to help with the broader regional scale reef degradation and loss, structure loss problem. And we just don't know that at this point.
Okay, thanks.
There's more that we don't know than we do know. So.
Right. Okay. So this question asks, I guess this is in relation, is there any particular species of seagrass? I'm sure that there are, so can you just mention some of the species of seagrasses that this work has been done with?
Most of this work has actually been done with Thalassia in the warmer tropical regions and Posidonia seagrasses in the colder areas. And so most of the work that I'm familiar with, and actually the seagrass beds that I work on, are Thalassia seagrass beds or combinations of Thalassia, Halidulea, and Serengodium seagrass. And in fact, some of the work I'm doing in Tampa Bay on the ability of seagrass beds to buffer ocean acidification shows that these seagrass beds can locally elevate pH by as much as half a pH unit at any one given point in time. So it's actually a very effective buffer for ocean acidification. What we don't know is how wintertime seagrass dieback and oxidation of organic matter is going to affect that particular scenario. And so that's one of the things that we're trying to work on at this point. But those are the species that are of primary targets so far for that kind of research.
Okay, great. And Lou Burnett asked, he says that excellent presentation, Kim. How sensitive are dissolution rates to temperature? This may be particularly important as temperatures rise. And I know you addressed some of this, but I don't know, you may be wanting some additional information.
So that's also a good question. Coral calcification rates actually increase with temperature to a certain point. And then once that temperature threshold is reached, then you begin to see a decrease in coral calcification. So it's going to depend on what the local temperature is. Certainly if you are breaching those thermal stress thresholds, then both a combination of thermal stress from temperatures that are too high and ocean acidification are going to exacerbate that dissolution or decreases in calcification. You know, we don't know what that balance is between increasing temperature that promotes calcification rates along with ocean acidification. That's something we have to tease out through some of these experiments that we're doing in the laboratories.
Okay. Kim, I have a question for you. In terms of some of the larger commercial and recreationally important species like snappers and groupers, are you aware of any research looking at some of these sites being used as spawning aggregations and changes in those aggregation sites based on the conditions of the reefs?
I am not aware specifically of any that's going on, but that does not mean that it's not happening. I'm not as in tune with the fish population research that's being conducted, and I know it is being conducted, but I don't know areas where they're specifically looking at it in concert with these other kinds of parameters.
Okay, thank you. Thanks. All right, I think that takes care of the questions, at least the ones that I can see on my questions screen. So Kim, wow, fantastic presentation. Thank you so much, and great slides. So if we did not get to your questions, or if you think of ones later today, please send them to Kim, and I'm sure she will certainly answer those, and you can find her email in the control panel of the chat box. And I know that we had some technical difficulties. Some of you had some technical difficulties at the beginning of the presentation, and again, this will be archived, and you'll be able to get to it on the SOCAN website, so you'll be able to pick it up there. Direct any questions that you have about this seminar series to socan@socorro.org. We'd like to thank you for attending this State of the Science webinar series today. We welcome any feedback or further questions that you have for any topics for this webinar series. You can submit input by replying to the follow-up email that you'll be receiving from us. And again, the recording of this webinar and a PDF of the presentation will be available on the SOCAN website.
Thank you.
Our next State of the Science discussion will be the week of Thanksgiving, which will be Tuesday, November the 24th at 11 a.m. And Jeff Scott from the University of South Carolina will be discussing the potential effects of climate change on the ecotoxicology of pesticides and contaminants, implications for ocean acidification interactions. And you can actually, registration is already available on the SOCAN website. We hope that you'll join us. Kim, thank you again for a very informative presentation, and we'd like to thank all of you for your thoughtful questions and for taking the time to join us today. We hope to see all of you at SOCAN's next State of the Science conversation the